Welcome to another virtual poster presentation. My name is Michael Hyatt. I'm a senior scientist in the bioengineering department. Today I'm going to talk to you about a poster that I presented at the 2018 AACR conference. So why do we care about 3D cell culture or want to use phenotypic analysis? Recently, phenotypic analysis methods have seen a resurgence for screening applications. We will show an example of a drug screen of 3D cancer spheroids and show that image-based phenotypic measurements can be used to detect treatment effects. We will also show this can be performed with basic tools and procedures available to anyone. To generate our 3D spheroids, we used Microwell. Microwell allows you to aggregate multiple cell types into spheroids of any given size. Size control can be achieved simply by changing the input seeding concentration. Regardless of what size you choose, you'll get a uniform distribution of spheroids. To demonstrate this drug screening application, we seeded a well-known MCF7 breast cancer line into Microwell plates at a density of 500 cells per microwell. These spheroids were cultured overnight in serum-free mammocult media. After 24 hours, the formed spheroids were exposed to drug treatments via a 50% media change. The drugs used in this experiment were chosen to target each of the 3 cell types present in breast tissue, as it is not clear which of these cell types is reflected in the MCF7 breast cancer cell line. Specifically, tamoxifen is known to target the ER-positive luminal cell population, while DAPT and lapatinib target the milk and basal cell types, respectively. The effects of these drugs were initially determined through measurement of cell viability, and later by image-based assessment of spheroid morphology using ImageJ, an open-sourced and free image analysis software. Briefly, an array of 16 spheroids per condition were analyzed, and a variety of outputs generated, including descriptors of spheroid size, shape, orientation, and brightness. Figure 3 shows the changes in viability in response to treatments. Panel A shows EC50 curves, and Panel B shows the total number of viable cells in each treatment. Together, these graphs show that EC50 values of approximately 17 micromolar and 6 micromolar can be determined for tamoxifen and lapatinib, but no determination of an EC50 value for DAPT can be made. This may be because cells of the milk lineage are not expected to be present in the MCF7 cell line. In later experiments, the published EC50 dose for DAPT was used. Panel C shows representative spheroids from tamoxifen doses immunostained with ecadherin in green and counterstained with DAPI shown in blue. These images demonstrate changes in spheroid size, organization, and ecadherin expression that match the drops in cell viability shown in panels A and B. Panel D shows that even at low magnification and using basic bright field microscopy, changes in spheroid morphology resulting from cellular changes can be observed across the entire tamoxifen dose range. It also shows mild disruption of surface smoothness appearing in 12.5 micromolar treatments and moderate to severe disaggregation occurring in the 25 to 50 micromolar range. Figure 4 demonstrates the impact on viability of individual and combined drugs treated at or near their EC50 ranges. Panel B shows that the combined drug treatments have additive or combinatorial effects. For example, lapatinib and DAPT have minimal impact on their own, but have a greater impact together or in combination with tamoxifen. As we would expect, treatment with all 3 drugs has the greatest impact on cell viability. While the previous experiments relied on viability measurements to determine drug effects, this can be time-consuming when performed on large screens. Figure 5a shows that even simple visual assessment of treated spheroids reveals changes in spheroid morphology. For example, a comparison of spheroid area versus measured cell viability for all treatments shows a weak correlation. Here, all of the treatment regimes yielding little to no cell death cluster at the bottom right, and the treatments inducing increasing reductions in cell viability align to the left. However, the weakness of this correlation indicates that multiple factors need to be considered. To identify which image-based measurements were indicative of a response to treatment, principal component analysis is shown in Figure 6a. This analysis shows clear separation between the control group, shown in gray, and the 3 drug treatment group, shown in green. Overlaid on this graph are eigenvectors. The direction and length of these vectors indicates the contribution of each measured parameter in helping to separate those 2 populations. For example, vectors that run along the dashed line contribute minimally to the classification of control or treatment groups. In contrast, vectors that are perpendicular to the dashed line, such as circularity, labeled here circ, indicate a stronger contribution to the separation of the control and treatment groups. Using the results of the principal component analysis, we graphed various combinations of these parameters to try and identify combinations that would provide clear separation between treatment and control groups. In Figure 6b, we show 3 such combinations. For example, showing circularity versus area or roundness versus area shows a partial separation of the treatment groups that had impact on viability from those that did not. In graphing integrated density versus area, we can see a clear separation between the treatments that had a substantial effect on viability and those that did not. So these analyses show that using only cell culture images and image analysis, we can clearly identify an effect from no effect. This means that when repeating this experiment with drugs of unknown effect, we would be able to determine if there was a substantial viability effect based solely on image analysis data. In summary, Aggarwell is an efficient tool for the standardized generation of multiple 3D spheroids per condition. Traditional methods, such as the determination of EC50 or IC50 curves via cell-based assays, can be supported. Furthermore, phenotypic analysis can reduce the time and effort required to detect treatment effects in screening applications. And who wouldn't want that? I hope you've enjoyed this presentation. For more information on Aggarwell or any of the products used in this study, and for more virtual poster presentations like this one, visit STEMCELL.com.